about Drosophila is you can then study all kinds of neuronal mechanisms in an in vivo system. And basically, here's a whole bunch of different systems that have been used in neuroscience. And I'm going to be focusing, of course, right here on Drosophila. It's got a vast array of genetic tools, as well as easy electrophysiology and quantitative morphological testing, fast life cycle, cheap to use. You can get billions of them with no problem.
And they fill your coffee cup.
They fill your coffee cup if you're not careful. Set up traps, it's no problem. And remarkably, because of the conservation that happens evolutionarily, many of the things that you find in all of these model systems end up having implications for human health. And indeed, absolutely everything my lab has ever discovered about the molecule synaptotagmin, which is what I study in Drosophila, those studies have been repeated in mouse, and things work exactly the same way. Okay, so why use Drosophila? As I mentioned, synaptic proteins are highly conserved. It's fast, it's easy, it's inexpensive, and you can generate specific transgenic animals quite rapidly. You don't have to pay for housing your animals in the mouse colony, so again, cheap. And then, importantly for our work, you get to test the function of your protein in an in vivo synapse, or in an in vivo system at a live synapse. So you can do quantitative analyses of the synaptic morphology, the electrophysiology, behavior, and you can do both forward genetics as well as so you can figure out what mediates this weird behavior that you've just discovered in forward genetics, or how actually does this protein work using the reverse genetics. And so I just need to start out with how the heck did we ever get started using Drosophila? I mean, a fruit fly. So Nettie Stevens, who was in Thomas Hunt Morgan's lab as a graduate student, was interested in the role of chromosomes in sex determination, and she was just using various insects in the lab as her model organisms, and she discovers the sex chromosomes, and she noticed that eggs only have X chromosomes, whereas sperm, some have X chromosomes and some have Y chromosomes, and she postulated, well, maybe it's sperm that determine the sex of the progeny, so she combined identified eggs with identified sperm, and sure enough, discovers that if you got a Y chromosome, you end up with a boy progeny. And similar studies were being done at a concurrent time by Edmund Beecher Wilson. But importantly, she was in the lab of Thomas Hunt Morgan, and he's credited with the birth of modern genetics. And so he was actually trying to look at evolutionary pressures using rats as his model system. But Nettie was in his lab, and he saw these flies, and it was like, wow, that life cycle is only 10 to 12 days. This is going to be a better place to try and look at evolutionary pressure so that you could do many, many, many generations in a shorter period of time. In addition, males and females are very easy to identify, and the crossing schemes are easy. And so he's working anywhere when he notices, oh, what's that? We have a white-eyed fly. And so he did a simple crossing scheme, took this mutant that he noticed, crossed it to a wild-type female, and in the F1 generation, the mutation was gone. And then in the cross-2 of the progeny, and then in the F2 generation, the mutation comes back. So this is a quarter show this. This is standard recessive genetics, reaffirming Mendelian genetics, which, well, that's great. But the important thing that he noticed was that only males had this mutation, which started out in the male, and only half of the males had this mutation. And through these observations, he discovered sex-linked inheritance. And so, here's just your standard. If you end up using flies, you'll learn all about that. But this brought up his theory of chromosomal inheritance. And in addition, in his lab, they started mapping lots of different mutant phenotypes to the X chromosome. So sex-linked, white eyes is located on the X chromosome. And who was it? Somebody else had seen these crossover events in the microscope, and in the Hunt lab, they had noticed that, okay, generally speaking, all of these traits on the X chromosome all ended up together in the progeny, but not always. Occasionally, they would separate. So for instance, body color versus wing size, 17% of the time, those would separate. They wouldn't stay on the X chromosome. And he came up with the idea of genetic recombination happening by way of these crossover events. And it then became known, the distance of these genes apart became measured in centimorgans based on Morgan. Okay, so this work ended up with many Nobel Prizes from people coming out of his lab, but in 1933 he got one for his theory of chromosomal inheritance, as well as the development at the time of what was called new biology, which instead of just observational, he developed the concept of, no, we've got an idea of how this is going to work and we're going to test it. And that was, Nobel Prize. So, at any rate, another thing that's important here, if you're going to use Drosophila, is this whole recombination thing can be a problem if you're trying to keep track of a recessive mutation that isn't, or even a dominant mutation that isn't labeled with something that you can easily see. And so balancer chromosomes are chromosomes where instead of having the normal order of your genes, they're all mixed up, so that when two of these chromosomes come together, the two alleles can't line up, and it suppresses crossover. And so you can keep by using balancer chromosomes, you can keep all of these things on the X, or the 1 or the 2 or the 3. Drosophila technically have 4 chromosomes, but the 4th is teeny tiny and nothing's ever on it. Well, it hasn't happened for us or most Drosophila people, but at any rate, so you need to suppress this crossover event to make it easier to keep track of your mutations. Okay, so that's all well and good for Drosophila getting developed as a genetic organism. What about neuroscience? So back in 1944, Catch describes this shaking phenotype. So you used to anesthetize your flies by putting them under ether, and he saw some flies that was like, whoa, that's weird. They're shaking. So another cool thing about Drosophila, you find a new mutation, you get to name it, and he named it Shaker. But back in 1944, we still didn't really know too much about these things. And then in 1969, Kaplan, working in Southern California, reaches and finds 2 more loci on the X chromosome that have this shaking phenotype. And so, like I said, cool thing about Drosophila, you get to name them. So the one was already called Shaker. The second one he called hyperkinetic, too much movement. But he had a third one to name. It's like, well, what am I going to name this? Well, he worked in Southern California, and as he walked home each day, he went by a certain disco called the Whiskey A Go-Go. Oh, you can't hear the sound. I didn't hook up the sound. Shoot. he named it Ether a Go-Go. I love that. Okay, so, but that still didn't tell us what are these mutants being caused by. And so in the 70s, the Jan Lab came along and they developed some very nice electrophysiological techniques. This is from a third instar larva dissected open to show its body wall musculature. And they would poke an electrode into the muscle fiber, suck up the nerve, and stimulate and see what happens. So in the wild type, you get a nice EJB, a nice evoked response. But in the shaker mutant, and this is even in less calcium because it would go wild, a whole bunch more neurotransmitter is released, and it lasts a lot longer. And of course, at this point, they knew that the so-called A current was a potassium current required for the repolarization. And so they compared this to adding 4AP, which would knock out the function of these channels, and saw a similar effect. And multiple other, of course, experimentation was done, but sure enough, it turns out that the shaker gene clones for a main subunit of the potassium A current channel. Okay, so that's just an example of some of the electrophysiology that you can do. They did, of course, controls to prove it wasn't anything to do with calcium, et cetera, et cetera. And eventually, the controls to prove it wasn't anything to do with calcium, et cetera, et cetera. And eventually, the potassium channel was cloned out of Drosophila based on the mapping that had been done of the chromosomes. But now, of course, we have a complete genetic map. So you know exactly where your gene of interest is located. Okay, so how about synapses? You know, can you really use them? And in Drosophila, if you're interested in human disease, and this is a neuromuscular junction from a Drosophila larvae, and here is the presynaptic nerve terminal with its clusters of synaptic vesicles. Focus on a little dense blob of electron-dense material here, which is the active zone. We have docked synaptic vesicles. Here's the postsynaptic muscle fiber. And really, the exact dense material is a little bit different. It has a T-bar shape instead of just being this amorphous blob. But all of the molecules that are involved in synaptic transmission are the same at the Drosophila neuromuscular junction. And like I said, everything that we've discovered with synaptotagmin, which is the molecule that I study, it works the exact same way. So, but our life cycle is 10 days, and our organisms are super cheap to use. Okay, so here is just a picture of a third instar larvae dissected out. You can see, I hope, the body wall musculature laid out here. Here's the central nervous system, nerves coming out. This is a very easy preparation. It takes undergrads maybe 1 or 2 weeks-ish to learn it for immunohistochemistry, a little bit longer to get it just right for electrophysiology. It's handy that it just so happens that anti-HRP antibodies label all neurons, so that's an easy way using immunohistochemistry to show your entire nervous system. And again, this is a very easy preparation for immunohistochemistry. So we've used synaptotagmin antibodies here to show that synaptotagmin is highly concentrated at the neuromuscular junctions, as well as in the central nervous system in the synaptic regions specifically. So it had been postulated in, I guess about the 1990s, that synaptotagmin was a critical molecule involved in synaptic transmission. And so this is an example of using Drosophila for reverse genetics. So the shaker phenotype was forward genetics. They noticed a problem and went, what is the gene, tracked it down, and it was a subunit of the potassium channel. Here is reverse genetics. You have a molecule that has been implicated in mammalian systems as being important for synaptic transmission, but we want to know exactly how it works, and so you can go into Drosophila and quite easily mess it up and measure specifically what's going on. Okay, so let's just knock out synaptotagmin completely and see what happens. And here is a synaptotagmin null mutant. Most of them don't survive to adulthood, but you get the occasional escaper, and you might get a normal synaptotagmin mutant, and they're a tag coordinated. They can actually write themselves. If once they do, they tend to just sort of spread eagle and move as little as possible. Okay, so this is typical of a disruption in neuromuscular transmission, and so using our electrophysiology techniques, we can go in and see what does the EFIS look like in a synaptotagmin null mutant. And if you do that in a wild type, you get a nice evoked response, around about 40, 45 millivolts. If you do that same experiment in synaptotagmin nulls, the evoked response is only about 2 to 2.5 millivolts. So clearly, it's 95% decrease in evoked transmitter release. Now, the synaptic vesicle cycle is a whole cycle of things. So this doesn't really tell you exactly what synaptotagmin's doing, but it says synaptic transmission somewhere in that vesicle cycle is screwed up because synaptotagmin is located specifically presynaptically. Okay, so the EFIS, this is again a fairly simple preparation. We could have undergrads in the lab doing that. This is the electron microscopy. Here's a wild type, vesicles chock full in the presynaptic terminal, focused at active zones. In the synaptotagmin null mutants, you can see that that cloud of vesicles is basically missing, and there's a few left, and the few that are left are brought very close to active zones, but there's a specific decrease in the number that are actually touching the presynaptic membrane or docking. So you can say, using these quantitative analyses, both morphological out what's various deficits. However, it turns out that synaptotagmin is most likely involved in several stages of the synaptic vesicle cycle. So just knocking it out, yeah, it tells you this cycle. It's not rubber coated. I'll stay far away. This cycle is disrupted, but you don't know where. So the genetic strategy for trying to figure out the specific functions of synaptotagmin. So we verified the sequence homology because we are actually interested in what's going on in human, and it's high conservation. It's in the right place. We've characterized our null mutant. A key important step for doing genetics in Drosophila is once you know what your null phenotype is, you must be able to rescue it with the wild type. Genetics in Drosophila is once you know what your null phenotype is, you must be able to rescue it with the wild type protein, because if you can't rescue with the wild type, you're sunk. And that's actually in the cycle that I study for synaptic transmission and specifically vesicle fusion events. The snare proteins that mediate those fusion events, VAMP or synaptobrevin, is located on the synaptic vesicle, and you put the wild type back in and it doesn't rescue. So this process isn't appropriate for studying that protein. But with synaptotagmin, here's what happens in the null. If we put the wild type back in, we get a pretty good rescue. It's not perfect, but it's not bad. This is about a 70% rescue, so a good enough signal to work with. The EFIS deficit is completely rescued, and so we've got looking at point mutations, and it's important that the wild type transgene is used as your positive control, because otherwise you would be overestimating any deficits that you see. This is as good as it gets. Okay, so at that point, you're ready to make specific mutations, put them into the null background, and then we have these quantitative analyses of synaptic physiology and synaptic ultrastructure that you can use to play and figure out how proteins work. Okay, so there's multiple functional sites in synaptotagma that have been mapped. These are polylysine motifs in both the C2A calcium binding domain and the C2B calcium binding motif. Of course, there are some hydrophobic residues that are important for membrane penetration, and then re-seen when it was redone in mammalian systems. Here's the calcium binding residues themselves, and importantly, right next to this calcium binding residue is in this SDP-YVK motif is a proline residue that was implicated in a congenital myasthenic syndrome. So there was a P to L mutation in this site, and in every member of the family that was affected by this myasthenic syndrome, and in none of the members that didn't have the disease. And because synaptotagmin is involved, and they had muscle weakness and easy fatigue and a defect in their synaptic transmission, and so at the neuromuscular junction. And so this was an obvious candidate, but when you only have one family, you can't say that that's causative for the disease. You can't say that that's causative for the disease. And so one of my graduate students introduced this same P to L mutation into Drosophila to see if she could reconstruct some of the deficits seen in people and then say, yes, indeed, this mutation is the cause of the congenital myasthenic syndrome. And so I'm just going to zip through these because I'm running out of time here. But at any rate, this is just an example of the electrophysiological recordings that you can do in the humans. It's a recording from the surface, the compound muscle action potential. And in these patients, it's smaller than expected. But if you have them do a maximal contraction, you get this facilitation, which the clinicians will tell you, well, that's typical of this in Drosophila, and this is just to show that when we took the heterozygous background and added one copy of the same mutation that the patients have to, because these patients were heterozygous, they had one good synaptotagmin and one bad synaptotagmin. If you have two of these in the fly, that's really bad. It's lethal. But with the heterozygous background, this is just to show our protein expression was appropriate. Half was coming from the transgene. It's sent to the neuromuscular junction, just like the wild type protein. And then when she did the physiology, sure enough, she did see a decrement in the evoked response. And so showing that there is indeed a that this specific point mutation disrupts synaptic transmission. But what we were really interested in was that increment that you would see after maximal contraction. And so she stimulated it at 10 hertz, and while we've normalized these now to the first response, just to emphasize that the wild type shows a decrement in this level of calcium, so less vesicles are available for fusion at future responses. But because these let out fewer vesicles in the first place, they don't show that depression, and so you do have a relative facilitation with high-frequency stimulation. So that's similar to what you see in the patients. However, a minute later, it had totally recovered. So that long-lasting facilitation didn't show up at 10 hertz. She tried 50 hertz since that's the estimate of how fast the neuromuscular junction is going during a maximal contraction in people. And she did see, again, facilitation during and right towards the end. It lasted for a couple of seconds, but by even 30 seconds, it was totally recovered. So the long-lasting facilitation, we couldn't recapitulate in Drosophila, which is really sad because that's the part of the disease that we just can't figure out what's mediating it, but we can't study that in Drosophila. What about the weakness? So there's behavioral assays that you can use in Drosophila. So this is a Drosophila activity monitor. Males and females have different circadian rhythms, so you have to separate them. And so she looked at the mutants, and basically during each of the phases of their activity, there was a significant decrease in the mutants, again consistent with a weak overall weakness. And fatigue was another, that's what actually bothers the patients the most, is the fatigue. Well, there's a wall climbing assay that you can do in Drosophila. Just so happens when you knock them down, they want to climb up. So what Mallory did is she would knock the flies down to the bottom of the vial, wait 5 seconds, and take a picture. And she'd do this every 10 seconds for 30 minutes. And this is an example of after the first drop down, and the wild types are mostly up at the top, and the mutants after that first one up, and the mutants after that first one, you know, they're not perfect, but they're doing pretty well. So they're a little bit weaker, but by the end of the 30 minutes, many, many more of the mutant flies are down at the bottom compared to wild type, indicating that they keep having to try and climb that vial. They're getting pooped. So both in the females and the males, we saw a deficit that was consistent with fatigue in the patients. So we got a similar decrease in muscle response following a single stimulation as seen in the patients. We saw facilitation upon high-frequency stimulation, similar to the patients. However, it didn't last the prolonged period of time. And we also were able to mimic the behavioral deficits showing weakness and fatigue. Drosophila studies were then good evidence that this point mutation in the family was involved in the etiology of their disease. And so it's a fast, cheap system for looking at a whole bunch of different, what I think, really interesting questions in neurobiology. And here are some of the folks. This is an older picture, but it shows the people involved in this work. So here's Matt, and here's Mallory, who were graduate students in the lab that conducted most of these experiments with the help of some previous undergrads, Katie Red, Laura Paranet, and Mackenzie Bolster.